Hey science fans, we're going to be looking at a triple topic today, helpful for the trilogy as well. So this is AQA paper 1, and we are going to be culturing bacteria or growing bacteria, bacterial culture in the lab. How we do it safely and cleanly, but also it kind of outlines how we grow microorganisms. Now we tend to use bacteria because it's quicker. It reproduces by binary fission very, very fast at low temperature, lowish temperatures, so we can get a large amount. There's a lot of keywords to learn, and then later on we're going to look at things that kill bacteria. So today you need to be able to do write out the stages of how we grow bacteria in a lab. Let's get cracking. Okay, first things first, we use these to grow our microorganisms in. So this is called a Petri dish. These ones are made of plastic, but they also come in glass. And the first thing you need to do is to sterilize it before use. So I'm going to write it on here. Sterilize Petri dishes. Okay, and we're probably going to do that if they're glass dishes, we would heat them up in an autoclave, which is a special oven. So if you hear the word autoclave, now these plastic ones come in a sealed packet, so we don't need to sterilize them because they were before starting. I should be wearing gloves, but we've already done this process, so it doesn't matter. We can also sterilize these by putting them in UV, so we can put them under a UV lamp, and that kills a lot of microorganisms. And then what we do next is we pour in this material. Now you can see a line here. Now this is a gel or a jelly. You can see it's quite shiny, and it's called agar. Now agar is what we call our culture medium. So that is where our bacteria is going to grow, or cultures. So they are going to grow on top of this agar. Now what the agar does is it produces nutrients. It's nutrient rich, so that the bacteria has, enough food, etc., enough nourishment. And this comes as a liquid, and it's like jelly. So you heat it up, it becomes liquid, and then you pour it in. Okay. So I didn't write this down. So our next stage was to pour in agar gel, which is our culture medium, and allow to set. Boom. Step 3 is going to be inoculate. And this is the part that has some quite important things. So when we inoculate, that means we are putting our microorganisms into or onto this agar plate. So while we're inoculating, you'll notice that I have a Bunsen burner present. So this Bunsen burner is going to be killing any microorganisms that are around this area, because obviously, fire. So we would do this as quickly as possible with the lids off. You can see the lids are off. There's the shiny medium there, the agar. And I'm doing this as fast as I can, but we will always have a flame present to kill any of the sort of yeast and bacteria that's no doubt in the air. So if I was going to inoculate with my substance, I would use an inoculating loop. Now, you're probably used to seeing these ones used for the flame tests. Now, to sterilize this, I quite simply just pop it in the flame, and that's going to kill any bacteria that's on there. Okay. And then I'm going to get my culture that I want to inoculate onto these. So the culture that I'm going to use is an E. coli. It's quite a safe one, although I'm not going to lick it. So I would like to inoculate my Petri dishes with this. So what I'm going to do is I'm going to use my sterilized inoculating loop, and I am going to put some on my sterilized inoculating loop. This is another type of inoculating loop. You can see it's made of plastic, so I wouldn't heat it to sterilize it, but it has been sterilized in these packets because they're individual use packets. So that's just to show there's more than one. Now I've got two, I'm just worried my phone's going to set on fire. I have got two types of E. coli. I've got a liquid one, and I have got this sort of solid gel type. Both of them have been come from sterilized culture containers. So again, I'm going to inoculate. I'm not going to inoculate. I'm going to sterilize my inoculating loop, allow it to cool down. Put my inoculating loop in E. coli. And then I'm going to very gently, so I don't pierce the surface, just rub that across the agar jelly, and then just really gently in a zigzag pattern across. So just gently in a zigzag pattern across the agar. I'm not prodding, I'm just wiping, because if I prod, I'm going to break the surface. Whilst this isn't dangerous bacteria, please be aware that bacteria can mutate very quickly for a change in DNA. So we need to make sure that we seal it. Okay, and then we have to tape it. Just left my tape over here.
With tape. Notice how I'm not sealing around the edge. I'm only sealing over the top because there is, realise I've just put, let me just use this one that I haven't used before. So you might notice that there is a little lip here. You can just about see it, there's a lip here. That's so oxygen can get in so that the bacteria can still aerobically respire. Whereas if I was to seal around the edge, the culture would just die because they wouldn't have any access to any oxygen. Now obviously, there is a limited supply of nutrients there, so they won't last forever, so they can only be used for our research purposes. Okay, just so you're aware, another way you can use these is you can actually grow, and this is what they do when they take bloods, etc., to see if you have any disease. You can grow culture mediums from people. So what I'm going to do is I obviously wash my hands, etc., but I haven't in the past few minutes. I'm just going to rub my finger along there, and then we're going to see if I have got any bacteria growing on there. So step 4 is incubate. So we're going to probably be putting those at around 25 degrees. We're not going to go much hotter than that. We don't want to grow anything really dangerous pathogens, like body temperature ones. We don't want harmful pathogens, so we're just going to do 25 degrees. We're inoculating, which we need to make sure that everything is sterilised. And that's it. They're the 4 steps of making a culture in the lab. Just going to... Oh, sorry, that's my finger over there. Just going to test my computer screen, my computer keyboard. I should imagine that'll be suitably disgusting. So there we go. I'm also going to do my mobile phone. So I'm going to do the buttons. I'm doing it right now while I'm rubbing it across the screen. So I'm just going to incubate these ones. So there's my index finger, there's my E. Coli. I've got my other E. Coli as well. That one's been on my nose, that one's been on my phone, and that one's been on my laptop. So we'll have a look and see how rank they look next time.
Left them for a week. And here we go. Look how gross. That is my nose. Got some good fungi in my nose apparently. And there's a really bit of colourful something bacteria there, which is bad. Colourful is bad. That's just my finger. Gross. Phone pretty gross. Mouth, not too shabby. Oh no, that's in mouthwash. That's a lie. Hand soap.